Hello there, dear viewer. Today, we're going on a thrilling journey that intersects gene therapy, machine learning, and artificial intelligence. Our goal: to harness ChatGPT's capability to rapidly prototype an AI system for producing and evaluating viral vector genome sequences. Yes, we're steering into the realm of democratizing biopharma solutions. I trust you have a basic understanding of the concepts at play. If not, buckle up and enjoy the ride. Our adventure begins not within the sterile walls of a high-tech lab, but right here amidst the vibrant world of citizen science. We set out with a dream to generate synthetic DNA sequences using AI and swiftly evaluate them for worthiness, without needing a lab coat. Our source of inspiration: GenBank, the trove of genetic sequences available to all who dare to dive deep into the genome. Our heartfelt thanks to GenBank for opening up this vast sea of information. On this quest, we're accompanied by a dynamic duo of AI, a Generative Adversarial Network or GAN, and an Artificial Neural Network or ANN. These models, our brave companions, were trained on real DNA sequences obtained from GenBank and shaped to create and assess synthetic DNA sequences. The essence of a GAN is akin to an artist-critic pair, perpetually engaged in creating and refining art. Here, the art is DNA sequences, and the critics are, well, tough to please. Navigating the labyrinth of DNA sequences proved to be a bit more complicated than our first hasty sketches. Our initial model produced more abstract art than Da Vincian masterpieces. And not all abstracts are Picassos, are they? Especially in the biological realm, where a single misplaced line, or nucleotide, can be the difference between a successful sequence and an unworthy one. On our journey, we face challenges. Optimizing the GAN's parameters is like solving a 4D Rubik's Cube while blindfolded and hanging upside down. It's complex, non-linear, and not something you'd want to do on a Sunday afternoon. Yet, we persist, because the implications for gene therapy and beyond are too vast to ignore. Acknowledging our trials and tribulations, it's clear that our GAN, while an enthusiastic learner, might need a sharper understanding of what constitutes a successful sequence. To address this, we introduced an ANN to evaluate the quality of the sequences generated. Trained on the same sequence data from GenBank, along with randomly mutated sequences, our ANN learned to differentiate between the successful and the not-so-successful with around 75% accuracy. Not bad, eh? It is important to note that the model may not have been trained on the best data. However, our journey wasn't all smooth sailing. We battled the tempest of errors, struggling with the context awareness of ChatGPT, and wrestling with potential misconceptions about the GenBank entries. We learned, we adapted, and we emerged stronger, armed with a better understanding of the systems we're working with. The results from this hypothetical AI system were, well, disappointing. When the ML models were used in a system together, the ANN determined that all sequences produced by the GAN were unworthy. This implies that the parameters of the GAN and ANN are both need to be optimized. However, I am not a machine learning expert, and this process cannot be easily automated. I would need collaboration to bring this project into fruition. This journey exemplifies the immense potential and challenges of applying AI in biopharma and gene therapy. AI can be an invaluable ally, but only when we understand it as much as it understands our data. If not anything else, we showed that a GAN can be applied to sequence data in order to generate synthetic sequences. We're just not confident on the quality for the purpose at hand. ChatGPT can be our research assistant, providing rapid prototyping, but we need to bear in mind its limitations and guide it with prompt engineering and explainable AI. Throughout this project, we maintained our faith in the power of citizen science and the potential of AI to change the world. We'd like to extend our gratitude to the teams at OpenAI and GenBank, without whose resources our voyage wouldn't have been possible. As we explore AI's potential to revolutionize biopharma and gene therapy, we must tread ethically and responsibly. It's not just about creating art, it's about understanding it, too. Thank you for your time. If you enjoyed this content, don't forget to comment, like, and subscribe. Also, feel free to check out the project GitHub code in the bio. There you will find more information on the methodologies used in this quick citizen science study. Here's to more adventures in the exciting intersection of AI and biopharma. Until next time, have a great day.